Hello everybody, this is Andy from Med School EU, and we're going to continue on with some educational videos on reproduction and inheritance. And the topic of today's video is going to be the genetic code. So we're going to take a look at gene expression, what is the purpose of genetic material, and we will analyze the first step of gene expression, which would be transcription. So first, let's discuss the purpose of genetic material. As we have these genes, we have, of course, in the last video, we talked about the general nucleotide structure where it has its phosphate, it's got the sugar group, and it's got the nitrogenous base. Now, the nitrogenous bases are going to be alternating between A and T and C and G, and they will be combined in the DNA double helix. Now, you must know that there is single-stranded DNA, especially in viruses. There's double-stranded DNA, just the regular DNA that we have in animals and plants. And there's RNA, and RNA is typically single-stranded. So these are all the different kinds of genetic material that would encompass all of viruses. However, in more of animals and plants, we would have just a double-stranded DNA. And also prokaryotes would also have double-stranded DNA, but not all, but most of them would have double-stranded DNA, except it would just be a circle and a single chromosome instead of multiple chromosomes. And so what is the purpose of all of this genetic material, and why do we have it inside of our nucleus, and how come prokaryotic cells also have genetic material? And typically, in order to have a live organism, something that is organic, it must have genetic material of some sort. So what is the purpose of it? Well, the whole purpose of DNA is to make proteins, and it is used to synthesize proteins. It's basically just a code for enzymes to be able to translate into proteins that would do functions for the cell. And depending on the cell, its location in the body, or its location in the organism, or if it's a prokaryote, its location in its environment, it would need to make different sorts of proteins to, for example, metabolize different products. If it has, for example, glucose inside its cell, it's going to need different enzymes, which are proteins, in order to metabolize that. And how is it made? Well, it's made through transcribing and then translating our genetic material. So we're going to take a look at the central dogma of biology, and we will discuss all the necessary steps in order to go from our DNA to our protein. And there are two major steps that happen. It would be transcription and translation. And in this video, we're going to take a look specifically at the molecular steps of transcription. So we're going to begin looking at our DNA, and this is just a DNA strand. Of course, as you can see, DNA has ATCG without the uracil, so it's got the 4 nitrogenous bases. They have complementary base pairing over here, and the importance of knowing the DNA and how it is structured and its anti-parallel and the way it's combined with its nitrogenous bases, having their hydrogen bonds in between. It's important to note which strand is going to be going 3' to 5' and which one will be going 5' to 3', and the significance of them in terms of their gene expression. So here what we have is the top strand. I'm going to have it going in the 3' to 5' direction, and I will have the bottom strand going 5' to 3' direction. So of course they're anti-parallel over here, and they're complementary, so AT, CG, CG, and so on. So this is our DNA molecule, just the DNA strand. There's only a couple of base pairs. Now moving on to the next step, how are we gonna get a protein coded out of this a coded protein out of this DNA molecule. So of course we're going to go through the first step of central dogma, and that's going to be transcription, which will give us our single-stranded mRNA molecule. So again, this is transcription. And in this section, this is going to be going in the 5' to 3' direction, and we have our mRNA molecule. mRNA stands for messenger RNA. So what's important to recognize is the ends here, 5' to 3'. So we're always going to have the mRNA in the 5' to 3' direction. So of course we're not going to be using both as the template strand in order to get to our mRNA. So we have two different strands here. One will be used as a template, and the other one will be just the coding strand. So 3' to 5' will be the template strand. So that's the template, because it's basically just a template for the mRNA. As you can see, UGG combines with ACC, and CCU combines with GGA. So this is complementary to the 3' to 5', and that's always going to be the template strand. Now this side is going to be the coding strand, or non-template. So we call it the coding strand. And why is it called the coding strand? Because this strand is exactly the same as the mRNA that's being made, except we replace the thymine with uracil. So sometimes with questions on the IMAT exam, you get questions that will give you the template and the coding strand, and it would ask for the mRNA molecule. Well, the easiest way to get the mRNA molecule is to simply look at the 5' to 3' strand, which is the coding strand of the DNA. And it's going to be exactly the same. You just replace the thymine with the uracils. And as you can see here, it's exactly the same. It's matching. So this mRNA is complementary to the template strand, but it is going to be the same as the coding, except for the thymine replaced with uracil. And we have our mRNA. Now typically this mRNA is made in the nucleus for eukaryotes. However, in prokaryotes they don't have a nucleus, so it's all going to be made in just the cytosol. Now moving on to the next step. After transcription, we have our translation. And translation goes to be a little bit more complicated than this. However, I'm just going to give you a quick overview of translation, and we're going to take a look at more of the molecular basis of translation in the next video. So this, of course, is translation. So we're going to be translating the mRNA molecule into proteins. So again, this is our mRNA strand. I just copied it from here. And some interesting things happen here. So first, let's label our 5' to 3'. You must know that the mRNA will always be translated in the 5' to 3' direction, and the protein, so the protein sequence or the peptide, is going to be made in the N' to C' direction. Just remember those things, and once we get to translation, this will be a little bit more clear. So what are these little alien things floating around? These are actually tRNA molecules. So they're also RNA. It's just a sequence of RNA that has the anticodon. So this is called the anticodon, because it is going to be complementary to some other specific codon that's on the mRNA. So for example, AGU is complementary with this one, and this is going to be the codon. So codon will combine with the anticodon, and there's going to be an amino acid, so that's amino acid, bonded to the tRNA. So what actually happens is there's a ribosome. this is the ribosome. And typically these ribosomes are going to be floating freely in the cytoplasm. They're going to bind to the mRNA molecule, to the 5', and they're going to begin their translation. And in order to read the open reading frame, they're going to need tRNAs to bind to them. So they're also going to come from the cytoplasm. They will bind to the ribosome, which will have 3 different sites. We're going to take a look in more detail. But what happens is they will come in, they will bind to their complementary codon with the anticodon, and it will release its amino acid and attach it to the ribosome. Now the ribosome, as you can see here, is going to build a peptide of amino acids. They're going to be bonded to each other in the N' to C' direction. And that's how our primary structure of a protein is going to be made. Now, of course, this protein has to undergo plenty of modifications in the rough endoplasmic reticulum and in the Golgi apparatus. So it's going to go through lots of processing before it is made to be functional. However, this is how it's going to be made and how we're actually going to get a protein, the primary structure of a protein, out of the DNA molecule. And that's the whole purpose of genetic material. So before we take a look at some of the specifics of transcription, I wanted to go over one of the most common questions that would be associated with genetics on the IMAT exam, and that is going to be just translating or transcribing a sequence of DNA, mRNA, and tRNA. You must be very proficient at exchanging between the codon, the anticodon, and the DNA sequence. So I'm going to show you just one quick little trick so that you are able to solve the question quickly without any hesitation. So what we have is a question that says, what is the tRNA anticodon sequence of the following DNA sequence? Now, this is the 3' to 5' DNA sequence, and we're going to go the long route in this video. And later I'm going to show you a quick modification that you could make to come up with the solution much faster. So we go 3' to 5', so we already know that this is the template strand. And what we're going to do is make our mRNA molecule first. So here we go. We have our mRNA molecule that's been transcribed. So of course this is going to be transcription, and as you can see, this is going to be complementary to the template strand, as we have mentioned earlier. And now we're going to make our tRNA anticodons that would typically bind to these codons of mRNA in order to compose their amino acids in a sequence. So this is our final tRNA molecule, or the tRNA anticodons, not the molecule, it's the anticodons. So let's label it anticodons. And they will be going 3' to 5' in order to be complementary to the mRNA. Now, so if you are asked the tRNA anticodon sequence, which would be this as the answer, you might think, hey, look, the tRNA anticodons are actually the same as the template strand, except you replace the T, the thymine, with uracils. And that's exactly right. So you could have skipped a huge chunk of a big step in transcribing it and then translating it to just simply replacing the thymine with the uracils. And that would have saved you plenty of time on the exam. So again, here, TAC, UAC, GCG, GCG, it's exactly the same on the tRNA anticodons. Now looking at the next part of the question, and it asks, what is the amino acid sequence that's going to come out of this transcription and translation? Well, first we're going to talk about something called the degeneracy of the mRNA codons. And this simply states that the third position on each mRNA codon is going to be called a wobble position. And it's called a wobble position because it's not as important as the first two positions. So let me explain here. We have three nucleotides, or three bases, I should say, that would be coding for a specific amino acid. Now, the first two positions, so this is 1, 2, and 3. So the first two positions are going to be strict. They're going to be very important positions, and they're going to determine pretty much what amino acid you are going to get. And the third position is called the wobble position because it's not as important. So for example, if we're looking at amino acid serine, what we have is you must have the first position as U, so you're going to put uracil. The second position is going to be a C, and this already determines that it's going to be serine. It doesn't matter, as you can see, it doesn't matter if it's uracil, cytosine, adenine, or guanine in the third position, because they're all going to be coding for serine anyways. So it doesn't matter which one is going to be, because it's going to code for it, and it's going to come out to be a serine. So that's called the degeneracy of the genetic code, and the significance of it is that it minimizes the mutations that we may have in the process of transcription and translation. Because if we, let's say, have a mutation here, a base pair mutation from a uracil to a cytosine, well, it doesn't matter. It's still going to code for the serine, and the protein is still going to be exactly the same because the amino acid has not been replaced. So we're going to use our mRNA sequence in order to come up with the amino acid sequence. Again, the mRNA will be going from 5' to 3', and the amino acid is going to go from the N' to C'. So first we're going to begin with the N'. The first part is the AUG. So AUG is typically the start codon for every single translation aspect. Now let's find AUG. So A is the first position, U is right here, and AUG is going to be methionine. So we put methionine, met. That's the amino acid that will come out from the AUG. And moving on. So this would be the final amino acid sequence, as this UAA is actually coding for the stop codon. So as you can see, U, A, and A is going to be the stop codon. So then the GGG, which would be GG, and G would be glycine. It's not going to be part of the amino acid sequence because we have a stop codon. So this would be the final amino acid sequence that would complete this question. All right, so now let's quickly take a look at transcription, and we're going to begin with transcription initiation, and this is typically what happens in eukaryotes. So this would be eukaryotic transcription initiation, and it slightly differs with prokaryotes. However, for the purposes of this course, it's not going to be as important to know the differences in terms of initiation and determination. We're more focused on just the global aspect of transcription and translation for the IMAT. We're not focused on the specific molecular details. However, I just find this quite fascinating, and this is something that I've learned in my undergraduate. So I would love to give some context so that you just have a general understanding of the process. So in eukaryotes, we would have the promoter site, which contains a data box. And the data box is only present in about 10% of promoters, in eukaryotes at least. And we've got these enhancer sequences. So it's just a DNA sequence that is called an enhancer because that is where the activators bind to. So that's going to be the activators here. And this is where they bind to the enhancer sequence. And activators are going to, of course, activate transcription. And the way they do this is they will bind to the enhancers. So they'll bind right here to the enhancers, and this will cause DNA bending. So DNA bending proteins are going to bind around, kind of clump around in this area, and they're going to bend the DNA over to the promoter site. So the activators are going to be near the promoter right here, as you can see there. Now what's going to happen is the activators are going to recruit mediator proteins. So that's going to be the mediator proteins, this big clump of proteins right there. And something that's very important, it's going to recruit general transcription factors. We call them GTFs. And general transcription factors are typically going to be sort of the main aspects that drive transcription initiation and the formation of RNA polymerase II on the strand. And as you can see, it's going to form the final form of transcription initiation complex, and it's now going to be ready for RNA or mRNA synthesis. So this is typically how transcription initiation happens in eukaryotes. In prokaryotes, something different occurs in terms of their initiation, as they have different types of RNA polymerases. And but we're not going to be going over in detail about the prokaryotic transcription initiation, as it is not a big part of the IMAT exam. Now, looking at transcription elongation, this would be probably the most important part that you should take away from this video for your IMAT exam. Is that you must know that there's going to be the coding strand, which would be the 5' to 3', which we have mentioned earlier, and there's going to be the template strand, which will go 3' to 5'. Now, the transcription elongation, of course, we have our RNA polymerase that will be going along and unwinding the DNA double helix, and it will make its mRNA molecule in the 5' to 3' direction here, because it's using the template of the 3' to 5' direction. Now, something very important occurs here is that it forms this 5' cap, which is, I believe, called 7-methylguanosine. We're going to take a look at the 5' cap and the 3' processing more closely in the next video, as these structures are extremely important for the mRNA, because it's going to protect the mRNA from enzymes. So again, you must know that this mRNA is going to be released into the cytoplasm. And cytoplasm is full of different types of enzymes, especially exonucleases that cleave nucleotides from the ends. Now, these 5' caps are going to protect its mRNA from being degraded before it's translated. So we have to keep everything intact for our translation so that it does not get degraded. We're also going to talk about the 3' processing in the future videos. Now, we're going to briefly discuss transcription termination. And again, this is not a huge part of the IMAT. However, it's going to be a great learning experience so that you know exactly how it occurs. And this is going to be more specific to prokaryotes, as they have two different mechanisms for transcription termination. So first, the first mechanism that we will discuss is going to be when the RNA polymerase is going to reach the terminator sequence. And this sequence, as you can see here, it consists of an inverted repeat and the T-rich region on the coding strand. So this actually, the T-rich region is right here, as it's shown right there, TTTT. That's the T-rich region, and inverted repeat. So as you can see here, this is the exact inverted repeat along with the coding strand. So what happens is that the significance of these parts on the terminator sequence is that when RNA polymerase gets there, it forms this hairpin. It forms something called a stable hairpin. And it sort of jams RNA polymerase from moving on to the next nucleotide. And as soon as RNA polymerase stops its transcription, it's going to stop working, and it's going to dissociate into its separate parts, and it's going to release the mRNA molecule out from its site. The RNA polymerase is going to dissociate, and the DNA will rewind back to its regular shape. So that's the significance of this inverted repeat and the T-rich region. Now, another way that prokaryotes undergo transcription termination is they have this helicase protein called the Rho protein. And this Rho protein is actually going to travel up to the paused or the stopped, the halted RNA polymerase. As RNA polymerase reaches the let's say here, the terminator sequence or the termination sequence, and it's going to stop. And this Rho protein is going to travel upwards towards the RNA, and it's going to cleave the mRNA and release it into the cytoplasm. So that's the two kind of general ways that prokaryotes undergo their transcription termination. Again, this is not significant for the IMAT. However, it is good to have just a general knowledge of. So this concludes our video on the genetic code. So today we took a look at the overview of the central dogma of biology. And in the next video, we're going to take a look at more details about protein synthesis involving translation.